Stormwater runoff occurs during and after a rain event as rushing water runs over impervious surfaces and is unable to percolate into the soil. This runoff has numerous physical and biological implications that can be damaging to the environment and the animals that rely on the environment, including us. The moving water picks up oils, metals, and other pollutants until it reaches an outlet, typically in a natural body of water. In addition to pollutant concerns, fast-moving stormwater runoff can cause erosion when it reaches natural waters, as well as flooding issues. As urban areas become more developed, the amount of impervious surfaces increases. This leads to faster moving water and at higher runoff volumes since less water is able to be absorbed.
Environmental engineers have developed numerous techniques to combat this issue. By slowing down the flow of runoff and naturally filtering the water before it can enter another body of water, water quality is improved. The Bioretention Cell by the University of Maryland School of Public Health is an example. This bioretention cell captures runoff from nearby paved areas and treats stormwater by allowing it to be absorbed by natural vegetation and is then filtered through the infiltration of soil layers. Water that makes it through the filter runs through an underdrain to the cistern for storage. A solar pump enables this water to be reused for irrigating the School of Public Health's community garden.
We understand that the filtered water in the cistern is of better quality after passing through the bioretention cell, but we do not have empirical values to back these claims. We performed tests on the cistern water as well as samples from Paint Branch Creek and this university's tap water to measure 8 water quality parameters. We compared the 3 sets of measurements and compared them to EPA regulations and effluent data from the Potomac Water Filtration Plant. Total suspended solids is an indicator of water quality. High levels of suspended solids can harm aquatic life and are an indicator of other forms of contamination. We used filtration to capture the suspended solids on glass fiber filter paper and weighed them before and after drying at 105 degrees Celsius. pH could drastically affect aquatic life if it falls above or below the neutral range. It can also harm engineered water systems due to the corrosiveness of low pH water. We used a pH meter to test each sample. Additionally, we tested for electrical conductivity, an easily measured quality of water that depends on the concentration of dissolved ions. Conductivity can be correlated to total dissolved solids, another water quality parameter. Conductivity was determined using a conductance meter. Nitrogen in a body of water acts as a fertilizer and can lead to algal blooms. Algae deplete the water's dissolved oxygen supply and limit the amount of sunlight that can reach underlying aquatic plants. Nitrate ingestion can also cause detrimental health effects in infants, pregnant women, and the elderly. We tested for total nitrogen using a spectrophotometer and correlated the absorbance of our samples to the absorbance of a set of standard solutions containing urea. Phosphorus, like nitrogen, is a nutrient, and high levels of phosphorus in runoff will fertilize water at the downstream end of the stormwater system, resulting in algal blooms. The test procedure for total phosphorus involves creating standard solutions of known phosphorus concentrations, adding reagents to the standards and the samples, and measuring standard and sample absorbance in a photospectrometer. Heavy metals in drinking water have numerous harmful health effects. To name just a few, copper ingestion can cause vomiting, diarrhea, stomach cramps, and nausea, and is linked to liver and kidney disease. Lead ingestion can affect the nervous system, especially in children, and high zinc exposure can cause fever, nausea, vomiting, stomach cramps, and diarrhea. To test for the concentration of heavy metals, copper, lead, and zinc, inductively coupled plasma mass spectrometry, or ICP-MS, was used. Atomic absorption spectroscopy, or AAS, was also used to determine the concentration of lead.
The test results indicate that the effluent from the bioretention cell is cleaner than the river water from Paint Branch Creek. In some respects, it was even cleaner than the tap water. Bioretention has been known to successfully filter nitrogen, phosphorus, suspended solids, and heavy metals from stormwater. If bioretention cells can be incorporated in more urban development designs, the overall quality of water in the surrounding environment could be significantly improved, reducing stress on natural bodies of water, making them more habitable to aquatic life. With cleaner natural waters, it could even reduce the money and energy costs needed to treat the water to drinking water standards. Special thank you to Locke, who led our team.